I'm happy to introduce our second speaker for today. Victoria Lee is an associate professor of history at Ohio University. So thank you for joining us as well, and feel free to share your screen.
5 years after the COVID pandemic, microbes have mostly disappeared from public everyday consciousness. But the same period is precisely when microbes have become lauded as the life support system of the biosphere by experts and moved for the first time to the forefront of discussions about sustainability. Microbes are now a spotlight of climate change studies and policy development, promising everything from soil bacteria to help us grow our food more efficiently, to engineered microbes to eat up industrial pollution or produce clean energy, or in cow stomachs or oceans to help slow down global warming. Like artificial intelligence, they are seen as a new frontier of untapped knowledge and technology. There might be 10 to the 31 microbes on Earth, more than the number of stars in the universe, and the vast majority have not been characterized yet. A new scientific conservation movement for microbes formed this summer, the Microbial Conservation Specialist Group of the International Union for the Conservation of Nature, draws attention to the tidal shift from the calls for elimination that we saw during COVID to the recognition of not simply inevitable coexistence, but ours and the planet's utter dependence on microbes. But we do know that microbes are not like other targets of conservation, such as whales, butterflies, or any animal on this chart. Charisma is the term used in conservation to describe non-human species, especially large megafauna that disproportionately capture human attention. Whatever charisma is, microbes don't seem to have it. By definition, they are too small for the eye to see, and not only do most of us not think much about them, we don't feel much about them. My project is a manifesto to reimagine how we and society relate to microbes and propose some alternatives to the goal of absolute control or eradication. I come to this as a historian of microbiology. My first book was on the 20th century history of fermentation science, or the science of knowing microbes as living workers as much as pathogens. The idea for the manifesto originated in a workshop I organized back in May 2021 at the height of the pandemic here at the Paris Institute for Advanced Study. It brought together 13 experts from academia, the dairy industry, and hospital medicine to talk about the need for another mode to deal with microbes, not just of eradication, but that also allows us to recognize interdependence and ideally to manage it as well. As a starting point, the topics looked at how we interact with microbes in everyday life: food fermentation, nutrition, the evolution of cheese, the human body and the microbiome, and antibiotic resistance. The manifesto will take that workshop as the seed and grow into a collective project. It will be an ongoing process, a document that will grow and change with collective input. As such, I openly invite ideas, suggestions, and contributions from anyone interested in the question. How can we reimagine the ways that we individually and society as a whole relate to microbes? And especially, what are some needed alternatives to the approach of absolute control or eradication? And now I would like to explain my own historical perspective, simply as a beginning to look at resources for creating the manifesto and indicating some of the directions it might go. It's by no means the product or endpoint, but a starting perspective. And I want to emphasize again that the manifesto will be left open to collective input, since this is, again, emphatically a collective project. Scientists often use numbers to argue for microbes' importance. We are about half microbes because of our microbiomes, and every animal and plant is a collaboration with microbes. We depend on them for our digestion, our immunity, perhaps even how we think. Oceanic microbes produce half the world's oxygen. Microbes are at least 15% of the Earth's biomass. Much of that is underground. If Earth's history were condensed to 24 hours, microbes appeared at 4 in the morning, which means all life has evolved to be surrounded by and deeply dependent on microbes. At a more personal level, there's been the microbiome health craze and the home brewing hype, but facts themselves do not always produce urgency. We are humans, and for humans, it's often stories that move us. So, for the rest of my talk, I will explain some of the stories that emerge from history as a perspective for why a manifesto is needed. Our powerful stories about microbes have long been focused on control, so much that microbial control has become an emblem of the arrival of universal modernity. Bruno Latour's classic study, The Pasteurization of France, sets Louis Pasteur's discovery of the germ theory of disease from the 1880s as the great divide between modernity and pre-modernity. It was not only the moment when science became the dominant authority to speak for the invisible world, but the moment when science gained universal control over nature through the laboratory. There was control over pathogens through sanitation and vaccines, and control over productive microbes in the brewing and fermentation industries. And yet, looking more closely at our history, the master story about control is a myth in several ways. First, the philosopher of ethology, Vincent Després, argues that it is not possible to learn about another living being if they are under absolute control. Knowledge about another being requires a degree of reciprocity, that is, mutual transformation or domestication. We each become a hybrid of the other. Clearly, when we artificially isolate and culture a microbe on a petri dish, the microbe becomes a with human hybrid. But human cultures, too, are with microbe hybrids. Our societies respond to disease, and we also build food and drug industries with microbes at the foundation. Thus, it is a myth that our challenges with microbes today are entirely unprecedented and that we have nothing to draw from our history. Fermentation practices trace historical continuity as much as discontinuity. There's a direct link between older sensory practices of selecting, preserving, propagating microbes and modern biotechnology. And today there's a deliberate push in science to draw on traditional empirical knowledge, focusing on certain charismatic microbes, such as Aspergillus oryzae, the mold used in sake and soy sauce brewing, or Penicillium camemberti or Roqueforti used in cheese production. Second, the insights of philosopher Baptiste Morisot, who writes about wolves reemerging in the French countryside, might also be applicable in understanding how we have imagined certain charismatic microbes historically. In the 1970s, smallpox was the exemplary charismatic microbe. It made us feel urgently about it, though not in a sympathetic way. Respected, feared, othered, and the target of a global eradication campaign by vaccination, smallpox was eventually declared eradicated by the World Health Organization in 1980. Morisot points to the fundamental myth that wolves, or in our case, microbes, remain wild, and that a boundary between us and them can be drawn and maintained. And yet the fact is they have co-evolved with us. We have each adapted our behavior in the constant presence of the other. Similarly, Michel Morange, the philosopher of biology, points out that for Louis Pasteur, at the founding of modern microbiology, it was unthinkable to challenge the boundary between humans and microbes, which is the very opposite of the microbiome revolution today. Yet, like wolves, microbes are still either bad targets of warfare or good specimens to be put in a sanctuary. Morrisov says we must move beyond this good-bad duality, which is premised on the assumption of progressive human control over nature. He asks, quite provocatively, can we recognize coexistence by having diplomats go between wolves and human society to negotiate mutually beneficial relations? He asks, is it possible to leave the wolves a share? Perhaps can we let the wolf take one sheep and the shepherd take the rest? And I'll come back to this question in relation to antibiotics, which is another story of human microbial warfare that plays out very differently from smallpox. The 1970s were a turning point in many ways. There was an ecological turn in the science of infectious disease and the rise of the broader environmental movement. James Lovelock and Lynn Margulis published their Gaia hypothesis, the idea that the Earth functions as a self-regulating system, and as part of that system, microbes are instrumentally important for all of life and for the Earth. These changes contributed to broadening arguments for microbial conservation. Between the 1970s and the 1990s, new developments in DNA sequencing technology that allowed identification of microbes in an environment without having to culture them, and new evidence on microbial evolution opened up a new perspective that prompted the biologist Stephen Jay Gould to advocate a reversal of our hierarchy of evolutionary progress. Gould argued that we should place microbes on par with humans, highlighting the diversity and complexity of their molecular systems, as well as their adaptability to different environments. In 1976, the historian William McNeill made a similar groundbreaking argument for microbial agency as a parallel to human agency in global history in his book, Plagues and Peoples. At the same time, in contrast to the history of eradication campaigns, agro-industrial institutions across the world were engaged in species-based microbial conservation throughout the 20th century, especially in the food and drug industries. These microbes are associated with an anthropomorphic charisma. We find them sympathetic and individual, almost as if personhood can be extended to them. Corynebacterium glutamicum is just one illustrative example of such a microbe. Discovered in 1956, it was valuable commercially because it is unusual in producing glutamic acid in large quantities. Glutamic acid is part of a family of amino acids and nucleotides. That is, they're the building blocks of proteins, DNA, and RNA. And some of these chemicals have a taste. And the ones that have a taste, they're now mass produced by microbes as commercial flavors based on processes pioneered by Japanese companies in the 1950s to 1970s. Our engagement with C. glutamicum highlights the valuing of biodiversity as much as standardization or management of contamination. It was found because scientists were looking for microbes displaying unique behavior. It was discovered in bird poop through a method to screen over 500 microbial strains in less than 3 months. Scientists were trying to capture a slice of the unknown diversity of the microbial world and use it creatively. It is a precedent for techniques like precision fermentation today, in which microbes are cultivated for their unique ability to do something. For example, fertilize soil in lieu of chemical fertilizer, clean up a polluted area, or make protein without animals or cheese without cows. Biodiversity is essential for industrial productivity. For example, during World War II, Japan scaled up alcohol production for biofuel substitution, and most of that research took place in the empire and not the home islands, because it was the diversity of microbial strains coming from heritage brewing cultures in colonial frontiers that was the source of the innovation. Similarly, small and medium scale French raw milk cheesemakers, who were often located in economically marginalized mountain regions, realized in the 1970s that they needed to start banking microbes to maintain the biodiversity required for production. And they were aided by state agricultural research institutes in the process. Such examples encourage us not only to think about preserving physical environments to conserve biological diversity, which the IUCN Microbial Conservation Group has advocated, but also about the importance of preserving cultural diversity and the role that state science might play in supporting social equity and small scale as well as large scale producers. Moreover, our development of C. glutamicum shows the prominence of ecosystem thinking as much as the individual organism. The microbe's biochemistry had to be re-engineered to manifest the desired behavior via human manipulation of the culture medium. But the human, too, had to be reconceptualized from a microbe's point of view as a consumer of microbial metabolites, in this case, the amino acid, glutamic acid, within a broader national ecosystem of chemical cycling. This kind of ecosystem thinking had a longer history in fermentation science, which used microbial food products to redistribute nutrients, such as proteins, amino acids, vitamins, or calories, within a national chemical ecology, especially in wartime. Techniques of ecosystem management to follow the flow of chemicals resonate with techniques today, for example, in studies of dairy farms that are involved in cheese production. And these studies attempt to follow the flow of microbes across the farm environment, the pastures, the soil, the guts or teats of the cows, the milking equipment, and eventually into the milk and finally into the cheese. In other words, not only our bodies, but our cultures and our epistemes have, to a degree, already been colonized by microbes to incorporate both the valuing of biodiversity and ecosystem thinking. Agroindustrial sciences are especially fruitful spaces for reciprocity. We know that soil microbes are, for the most part, unculturable by humans. For the vast majority of soil microbes, it is impossible to isolate them from their environment and grow them on their own in a petri dish. But cheese scientists note that domesticated microbes that humans already use in agro-industry are more accessible to us. A larger proportion of them are accessible to us through classical culturing techniques compared to wild microbes in the soil. Finally, to come back to the question of warfare, in the case of antibiotics, humans have domesticated microbes, beginning with the penicillin mold in the 1940s, to make chemicals for us to kill other microbes, while the enemy microbes evolve antibiotic resistance. This is an example of coevolution and domestication in ways that are mutually destructive. And it shows that the split between good and bad microbes in our minds is a misguided question. Paradoxically, we have had the genetic and ecological knowledge since the 1950s that high antibiotic use is clinically unsustainable due to resistance. But it was only since the 1990s that superbug cases provoked recognition of the need to restrict use. Why is that? It's because in a simple control paradigm, softer, more ecological approaches are dismissed as technologically regressive. And this is precisely why it is a paradigm we need to escape. Going back to the question of whether we can meet the microbes halfway or leave a share for the wolves, there are new proposed directions, not only in antibiotic stewardship, but also in phage therapy, and even in novel methods of ecological approaches to drug mining, in which we might learn to culture microbes not in isolation, but within the commensal or mutualistic relations in which they live in their natural environment. Even as microbes are especially invisible, especially intractable, and especially subject to myth, history shows us that the repertoire of human relationships with microbes potentially could be, and historically has been, wide. In response to Baptiste Morisot's lament, we are a species that tends to colonize by totally excluding others, risking the destruction of the biodiversity that sustains us. We could instead rise to the challenge of the Indigenous science studies scholar Kim Tallbear. I want the biosciences to be colonized, inhabited by those who were formerly only the food or subject for science. As we turn worldwide from efforts at eradication to movements for conservation, our history and our present offer possibilities for exiting the modern myth of absolute control, opening up instead approaches toward the valuing of biodiversity, the incorporation of ecosystem thinking, and the scientific support of social and cultural aspects of sustainability as we begin to recognize that we live inherently microbial lives. And with these possibilities in mind, I once again invite a collective response to the open-ended question that will be at the heart of the manifesto. How might we reimagine the way that we and society relate to microbes? And what are some needed alternatives to the goal of absolute control or eradication? Thank you.
Great. Thank you so much. Yes, Sadi, first question.
Well, thank you so much. I'm so glad to be the director of this institute. I mean, this is exemplary of the kind of important research topic and new approaches that we have here. And thank you for being heading this initiative of the manifesto. So, I mean, it's very clear that we cannot win a war against microbe. I mean, they evolve faster than us. And of course, they're part of us, as you said, and we're part of them. So what I find both interesting and it's also my question is how are we going to build that manifesto, right? What you showed here is really interesting. It's just like we have been doing some conservation of streams of microbes for various industries. History is actually keeping possibly the streams of what types of collaboration we've had with microbes in the past that was successful and those that weren't. And I was wondering if this historical approach could not be the start of what will come with the manifesto, which is the resource that we want to use to do this collaboration with microbes. And I was thinking of another great project that emerged here at the Institute, the one by Alena Ledenieva on the Encyclopedia of Informality, which became a huge international effort where all, well, hundreds literally of scientists, mostly, I mean, ethnographers, sociologists, wrote about a small piece about one type of informality that they observed in their culture. And this creates this huge online now encyclopedia, which now has 3 big volumes. And I was wondering if connected with this manifesto that you'll be writing while you're here this month, we could create locally, and that the Institute will be happy to host this, some kind of a repertoire of what are the ways we could collaborate with microbes, or we have done in the past. How do you think practically you're going to, I mean, to do the things besides this manifesto text, and how to push this collaboration worldwide?
Yeah, thank you. That's a very interesting suggestion for an encyclopedia of mutually beneficial relationships with microbes, and sort of conscious collaboration rather than we are already a collaboration, but we haven't been thinking about it as much as we should have. And having an encyclopedia that would actually document and reflect on this and have and sort of categorize it, make it a taxonomy as well. So that's a really interesting suggestion that hadn't occurred to me before, and I'm glad to know that there's already a reference point for that, because it is such a huge field, in terms of the potential areas that people could contribute. I mean, my own knowledge, it just comes from one small area within that, which is industrial science, applied in industrial microbiology in a specific context, Japanese fermentation. Whereas, there are huge communities working on, even just in agro-industrial science, there are huge communities working on that in STS of sort of cheese production in France, and within the AOP system, especially, and sort of a food history, but also other angles, medical angles, antibiotic resistance, or thinking about pathogenesis. So I see Pierre Olivier is here. What makes a pathogen a pathogen? Klaas Kerschel is here. I see Klaas Kerschel is here as well, and thinking about sort of how we manage this condition of antibiotic resistance. I mean, these are all huge areas, and yeah, so they're all ways to go to think about this. And Matthias Grote is here as well. I know he's working on a project on Lynn Margulis and Earth system science, so how microbes became important, seen as important as underpinning our entire biosphere, and kind of thinking about these metaphors of warfare. Yeah, just kind of a shout out as well to Ken Daimaru, who's here, like thinking about medicine and warfare, and how that, not just as a metaphor, but a metaphor that has really permeated our practices. and how we might be able to move beyond that duality of good and bad to think about how are microbes as they are, how are we as we are, and how can we understand that going beyond this very anthropocentric view of just are they going to make us sick, or can we exploit them to make money?
Great. Nitsa next.
Hi, thank you. I loved how you brought together issues of public health and smallpox eradication on the one hand and the joy of food on the other. But it also brought up for me the question of whether in the manifesto and in general you're interested in the global preservation of microbes, or are you actually interested in separating between the good ones and the bad ones, right? I mean, what is actually the project of preservation for you? Is it just let it, as you said, let the wolf roam? Or do you actually, are you, as a collective project, concerned with, for example, not bringing smallpox back, right? I mean, what are your political, I guess, boundaries here?
Mm-hmm.
And actually, that relates to the question of, does the manifesto also includes the means to preserve the microbes, right? I mean, the strategies, both political and or practical. I would love to hear you talking about that as well.
Yeah, thank you. And it's been quite a big topic of discussion because of this new microbial conservation group at the IUCN. They've been in the media talking about, well, we all know microbes are important, but how do we raise public consciousness of that, and how do we change people's minds in public? Because when we think of microbes, mostly we think of smallpox, and that's mostly it. So they're talking about microbes as having an image problem. And I'm very interested in that problem. So more than the physical preservation of microbes, which I think it would be useful to talk about that, because they require, historically, it's required a lot of resources. It's a very resource intensive, labor intensive, capital intensive endeavor. It's very costly to maintain microbial collections. And in fact, in the past, you used to have to reculture them. You had to take them out and reculture them every now and then, and just to make sure the same ones were still there, and that you didn't have something else that was contaminating it that you didn't know about. And now, I mean, there are sort of lots of suggestions for how, well, maybe we don't need to preserve the physical microbes. Maybe we can just take their genes and then preserve that. Maybe we can do that electronically or whatever. So there are kind of discussions about how to be resource efficient about preservation as well. But that is one of the big questions. How do you get the public behind it and to support that these efforts? Because it may seem very abstract or blue sky. Like, why are we doing this? Aren't we more worried that we're going to release some terrible microbe upon the world? And so it's about broadening public imagination of why microbes are important. And initially, some of the philosophical arguments for, I mean, it's very interesting because some of the philosophical arguments for conservation did revolve around smallpox. Like, should we destroy the last remaining stocks so that it never infects anybody again, or should we preserve them in case we need to research them? And that's, that's one specific area within a much broader picture. And I sort of want to get at the much broader picture as well. Thank you.
Great. Class next.
Hi, Victoria. Thank you. That was a really interesting and brilliant talk. I really enjoyed it. My question comes from, I guess, being a German, and always when I hear manifesto, I think of Marx. So, I think for me, the question with the Microbial Manifesto is also one of ownership of microbes and rights to microbes. So we've had over the last year 2 publications coming out on rights to microbes and potential microbial rights in themselves, but none of these publications actually raised the issue that there is no longer a microbial commons in the world following the 1992 Rio Declaration, right? So microbes are either sovereign, not necessarily property, but there are sovereign control over access and use of microbes. Nagoya has tried to resolve this, but kind of didn't really manage to do this. Lots of microbes, the ones that are valuable, if they're not patented, at least closely secured, and within industrial process secrets, they're kind of bound up in those. So in many ways, the microbial commons has become almost captured by a kind of race to secure microbial value. So I think the issue might not actually be not recognizing the value of the microbial commons, because I think that's actually become quite evident with the multi-billion-dollar industry that's evolved around it. If I think about biotech and patenting, etc., it might be more a question of how do we ensure more equitable access to these microbial commons, and how do we overcome very justifiably held biases or concerns by especially low and middle income countries about a return to a kind of like extractive, conservation enveloped discourse about securing and appropriating valuable microbiomes and really ensuring that benefits are distributed.
Yeah, that's a very good question, and legally probably very, very complicated. Just from informal conversations with microbiologists, I've heard that it has made their work a lot more difficult, because it's difficult to just sort of gather samples where you'd go to a tropical place, perhaps Indonesia, gather samples of the microbial diversity there, and then take it back to your lab and research that. And that's got very different implications now. But on the other hand, perhaps that also needs a bit more sort of clarity. What would it mean for people to be compensated, communities to be compensated from microbes that are taken from their area, or what does ownership mean? And microbes, they do become engineered and manipulated, and they become something quite different too from when you first pick them up. But yeah, they're very difficult, I suppose, and thorny issues. And yeah, I would agree that there are that, especially in the private sector, that the value of the microbial commons has been recognized for a long time, and in fact is increasing that recognition. So there you hear reports of big algae factories being built up, and and how algae are going to replace manufacturing in plastics and so on. So there's a big hope in some of these areas, sort of maybe even an over optimism technologically, but yeah, that's a difficult question, and I think a very important one. Thank you.
Great. And Sadi, yes.
Thank you. If I can add something, I mean, this commodification of microbes is actually part of the problem that we should be tackling. I mean, we have to reconsider the collaboration we have with the rest of our environment, and that's part of it. And precisely if there's no, if the, I mean, not the set of freedom of microbes, but I mean, the sort of importance of their existence per se, and not as objects for our human industry, is not recognized. I mean, we are going further in the type of exploitation of our environment that has led us to the dire situation where we are. And so I think there's a philosophical aspect in this. And what is it we do with the rest of our environment to be responsible and to be sustainable? And putting the light on microbes, which are currently being appropriated or used, is certainly something that should bring forward that thing as a political issue more generally on what are the types of production and exploitation of our environment that we're having. I don't say that because exploiting is bad per se, but it has very bad consequences if it's not reflected upon. And I think writing the manifesto and the discussions around it should be part of that discussion. And this is why we invited Victoria, because we think that she is working on something that is absolutely vital, I mean, for the planet and for our society.
All right. Yeah. Thank you. Yeah, I do think that the focus on which microbes are useful, which microbes are harmful, that has driven a lot of research, which in that sense it's been stimulating and it's been a positive, but one has to get beyond that a little bit. And you see that in the discussion, in fact, of antibiotic drug mining. There's often and the whole property of biodiversity that makes it valuable is that we don't know what it is, that it's unknown, that we don't really know what's out there. And in antibiotics, the discussion has been a lot around, well, part of the reason why we can't solve the problem of resistance is because microbes are not producing new antibiotics for us anymore. We've hit some kind of technical limit with what we can get out of them. But then you have scientists saying, well, if you just take a step back and think about, maybe antibiotics don't always act as antibiotics in their natural environment. Maybe they don't always kill other microbes. Maybe they're part of very complex chemical signaling systems that we don't understand very well. Maybe we should see microbes as they are and not simply as, in the anthropocentric categories that we put them in, like, antibiotic producers or targets of antibiotics, and instead think about, well, if we looked at how they live in their natural environment and understand more about their ecology, the way they fit into human ecologies, and in a more holistic way, rather than focusing on, kind of very direct or short-term exploitation, or pure culture, sort of direct pure culture. If we kind of take a more holistic view, a more ecological view, that might actually benefit us in the long term, including, for commercial purposes and clinical purposes.
Yes, Jeremy.
I'm shy to ask a question yet again, but this would provoke so many things for me. And I was thinking about the ways in which there's so many other modes of engagement with, food production, soil, and so on that are dealing with non-Western or non-hegemonic Western forms of agriculture. And as you were talking, I was thinking, I don't know if you know the work of Karina Treitel, the book Eating Nature in modern Germany, and she talks about the theosophists like Rudolf Steiner and others for whom who develop bioenergetic agriculture, but this is all sort of operating under the understanding that the soil is alive and it's spiritually alive. It's not just physically alive, and it makes me think about all of the kind of cosmological sort of dimensions of your call for this kind of forum of cohabitation and the ways in which microbes could be positioned in different sorts of cosmological frameworks. And I'm wondering if that's something that you're also thinking about, or does that figure in some way in the way you're imagining this new forms of relationship?
Absolutely. I think that's a very interesting suggestion. And there are people like Robin Wall-Kimmerer, other writers coming from Indigenous science studies, Kim Tallbear, who have long advocated that we rethink the way that we relate to other living beings. So it would absolutely be very interesting to draw from that sort of thinking about our relationships with the natural world and how there are many ways that we could reimagine those.
All right. Very good. This is all the time we have for this section. So thank you so much, Victoria, and everyone for your questions.